Okay, this is the IAA experiment in plant responses in your book. It's on page 369 in your book. So what we're going to look with this IAA experiment, if you will remember, IAA is indoleic acetic acid, and here is some concentrated dried version of IAA. It is a plant growth regulator or a hormone. And what we're looking to do is to see IAA, we know, is produced in the tips and the shoots of roots. It is also produced in seeds themselves. So we are going to be looking at a certain plant tissue, and the plant tissue we're looking at are cress seeds. These aren't cress seeds, but just imagine. This is the best I can do at home. So these are my cress seeds, and I'm looking to see what effect this hormone has on the growth of the roots and the shoots of the seeds. So I'm going to set up a series of dilutions, and I've set up my experiment here with my own version of Petri dishes and bottles and filter paper, cotton wool and lids of Petri dishes. So just go with me on this one. Okay, the first thing I have to do is I have to make up a stock solution of IAA. And when I say I mean the teacher. And I'm wearing gloves because IAA is toxic. So your teacher would always set this up. IAA in concentrated form is quite difficult to dissolve. So what we have to do is we have to dissolve it, first of all, in some alcohol. So I have some alcohol here, and I'm going to dissolve my IAA, and it's pre-measured so I can get the correct concentration of IAA. So that goes into my alcohol, and I'm going to stir it with my stirrer until it has dissolved. Okay, and IAA is difficult to dissolve, and that is why it's dissolved in ethanol. When it has dissolved, I'm going to add it to a graduated cylinder. This is a graduated cylinder, and I'm going to make up a stock solution. So I add my IAA to my graduated cylinder, and I'm going to make it up with, it should be distilled water, and we use distilled water so we know that it's pure and there's no other possible chemicals in it. And I make it up very carefully to a 1 litre, looking at the meniscus and looking at the graduation marks. So just go with me with this one. Again, I would give that a little stir to make sure that the concentration is the same throughout my stock solution. Okay, now to set up the experiment, what we have to do. You have got Petri dishes labeled A with a concentration of IAA solution of 10 to the power of 2, well, that's just 100 parts per million. Okay, so that is the most concentrated IAA solution that we are going to be working with. If you move down, we've got another Petri dish, B, with 10 to the power of 1 parts per million. And move down, you can see D is 10 to the minus 1. Continue down to a really, really diluted concentration of IAA with G, which is going to be 10 to the power of minus 4, 0.0001 parts per million. So it's really, really diluted. And at the end, we have a Petri dish with no IA in it, and we act that as a control. That is only going to be distilled water. Okay, so how did we set up the experiment? You get your stock solution, and again, you're going to have to use a graduated cylinder, and you're going to put 10 mils, as best as I can measure, 10 mils of IAA solution into your first bottle. So that's bottle A with 10 mils of your IAA solution. Now I'm going to start my serial dilution. To get going, what you have to do is, and just go with me with this one, again, you're going to use either a pipette or a graduated cylinder, and you're going to put 9 mils of water into each of these. So 9 mils of water, of distilled water, just imagine I'm measuring it really accurately, goes into the bottles A, B, C, D, E, F, including G. So we now have 9 mils of water into all of these, and in this one I have 10 mils of my stock solution of IAA. Now that I've done anything that's kind of concentrated, I can get rid of the gloves. I don't need those anymore. And students wouldn't need gloves because the IAA solution that you are using is so diluted it wouldn't harm you at all. Okay, I have to dilute this solution by a factor of 10 each time. So what I'm going to do is I'm going to use a clean pipette. Okay, this is my clean pipette. And I'm going to take 1 ml, taking up 1 ml of stock solution from A and put it into B. I'm going to use my pipette to stir my stock solution. Remember, there was water in here, and now I've added 1 ml of my stock solution to it. I give it a stir, or I can throw my pipette away now. I could put the lid on it and give it a really good, so I'm making sure that it's the same concentration throughout. When I've done that, I've thrown away this pipette, I get a fresh pipette. That is really important. You need a fresh pipette so that you're not contaminating your stock solution or your concentration of your diluted concentrations. You've got to get a clean pipette. Clean pipette, 1 ml of the solution that is now in B. Imagine I'm taking up 1 ml and I put it into C. I give C a stir, or I put the lid on C and give it a little shake. I continue getting a fresh pipette each time. Taking 1 ml from C, put it into D. Stir. Fresh pipette. 1 ml from D to E. 1 ml from E to F. 1 ml from F to G. And at this point you stop. So this could be some of the questions that they could ask you. Do you put any IAA into your last bottle? So I now have 10 mils in here, and it's really, really diluted because it's 10 to the power of 4. I have 10 mils in here, but I only have 9 mils of water here. So what I'm going to do, the last thing you do is you take 1 mil from your second last bottle and you just put it down the sink, and that's it. So now if you look at them, I've got my most concentrated IAA solution in A, and it's going diluted, diluted, and even more diluted. So this is called a serial dilution, all the way to E, which is really, really dilute, and H has got no IAA in it. It is the control. Okay, now we're going to set up our seeds. So to set up the seeds to see what effect the IAA has on the seeds, I get acetate. Now just go with me in this. Acetate is like a piece of plastic, and I photocopy grids, lines of them. And I put them into the bottom of my Petri dish. And into the Petri dish, I take 5 seeds. We are using cress seeds. You can use radish seeds. I spread them out and I put them into the middle, if you can see that. I put them into the middle of the Petri dish, okay? So I now have my acetate with my grids on it and my 5 seeds. The reason I use 5 seeds is we're replicating. It's important to have replicates to have repeat the experiment, and that increases the accuracy. So the reason you have this grid system here is it's going to make it easier when we're looking to see how much shoot growth and how much root growth we're going to get in the seeds. And all we'll have to do is count those squares instead of having to try to use a ruler. Okay, I have set up all of the other Petri dishes the same. So each of them have now got 5 seeds and they're all in the middle with our acetate. So what do we do next? We are now going to add our serial dilutions of the different concentrations of IAA to each Petri dish. So the first thing you do is you put down a small piece of filter paper so that it will cover the bottom of your Petri dish, and you add about half of your IAA solution. Okay, perfect, so that it is wet. The filter paper keeps the seeds in place. That might be a question that they ask you. So the filter paper keeps the seeds in place. Then we get cotton wool. Now, you need a lot more cotton wool, but that's all I have at home at the moment. And you put your cotton wool in on top of the filter paper. You add the rest of your IAA solution. And what the cotton wool does is that it makes sure that the seeds are in contact with the water or your solution the whole time. So it gives them a chance to grow. You do the same for each of the other Petri dishes. So I'm just going to do them very quickly. Filter paper, some of your water, about half your water, then your cotton wool, then the rest of your IAA solution. And you continue on. I won't do all of them, but you can get the idea. Okay, now that we've got all of our Petri dishes with their appropriate, the right concentration of IAA solution, what we're going to do now is I'm going to keep my label because I'm going to need my label. Stick it there, and I'm going to get the lid of my Petri dish. So just go with me on this one. This is the lid of my Petri dish. Now you have to keep an eye. I know my seeds are in a horizontal line like this in the middle, and I have to keep an eye on that because I have to keep the seeds horizontal. There should be loads of cotton wool in here, keeping everything in place. I put my lid on and I get some parafilm, it sometimes says, but just some tape. So we're just using a type of sellotape. And if I can do this, it's going to be a bit tricky. I'm going to seal the lid. And this just makes sure that everything is secure. So the lid is in place. Everything is secure. And that the cotton wool won't fall out and that my seeds are in place. Now you can just, it's not too bad. You can just about see across there are my seeds. They're in a horizontal row. So that means horizontal, that is obviously vertical. The filter paper and the cotton wool is keeping them in place. So now I want to see what effect my different concentrations of IAA have on these seeds. They're going to start growing. Shoots are going to grow up and roots are going to grow down. Not in all of the Petri dishes, but most of them. And we're going to see which ones grow better, which ones grow bigger. So what we do is we get all of our Petri dishes, and they're all going to look like this. And we put them into a container, making sure, this is perfect as well, making sure that they're horizontal this way. And we're going to leave them for about 2 to 3 days, enough time for the seeds to grow. Then when we're finished, we take them out and we have a little look and see what the growth is like. And we're going to compare everything to the control. So the control has no IAA in it. And the control, these seeds will have grown as well because the seeds produce their own hormone, their own IAA. Okay, so imagine I'm going to count all the little squares of all the different growth from all of my 8 different Petri dishes. So you'll have to go with me on this one. This is something sort of what it will look like. There is my acetate grid in the background. Here's my 1, 2, 3, 4, 5 seeds. I'm looking at this one and it's grown 1, 2, 3, 4, 5 squares. This one, the shoot has grown 1, 2, 3 squares, 1, 2, 3 squares, and so on. And I do the same for the roots, 1, 2, 3, 4, 5 squares, and I continue that way. I get the average, obviously, of all of those, and I record it in a table. And what we're looking to do is to see what the shoot growth and the root growth is in comparison to the control. That's what we're looking at. Now, all of this is way too much detail for your exam, and you won't need all of this for the exam. It's just so that you will understand what we're actually doing here. So we're doing a serial dilution. You've got to know that your seeds, which is our plant tissue. You've got to know about your IAA and dissolving it in ethanol or alcohol. You've got to describe as best you can your serial dilution. You've got to explain why there is acetate, why you have filter paper, why you have cotton wool, and you might need to explain about how you put your lid on and how you seal it and how you place it horizontally in some kind of a container to keep them horizontal, and know it's 2 to 3 days that you let the seeds germinate. You do not need to write everything that I have done there, and I'll give you an example of how you could write it out to get full marks.